Thank you very much, Fred. So I'm an academic, but I also look at companies, but I start in my academic lab, and I'm afraid I probably do it differently from the last speaker recommends. We can discuss that. So I'm interested in these things, which are proteins, and they provide rather nice targets for drugs. But I can't do everything in my academic lab, so another thing I do is to found companies. And this is Aztecs, and after 15 years we managed to sell it for a reasonable sum of money. And that reflects the cost of making drugs. I mean, we had 250 million in investment. And so the question that I've been asked to address is how we use the sort of traditional making of drugs with the target base to deal with situations like this. And this is AKU target, and what we find here is that we have lots of different kinds of mutations which are all classed as AKU. We can subclassify them, and I'll say a little bit about that. But of course the problem is that if we're spending huge amounts of money on each drug, and after all of that time we have a couple of drugs in phase 3 and one in phase 2 and a few earlier on, but we still haven't got anything on the market, how are we going to deal with this? And that's the theme, of course, for this evening. And I'd like to just make one point, and that is that the emergence of resistance poses a similar problem, because we have multiple mutations in a very small numbers of pathogens or patients, or patients really, and so that's the problem. And how do we deal with that? Well, I've got a very complicated flow diagram, but I'll come to that later. I'm going to try and not be too technical. We started on Aztecs, and I just described it. It's one of the companies I founded, and what we have is a target for a drug. We can draw out the drug and understand how it interacts. But of course, we normally start with the target, and we have to work out how to get from there. And so what we did is take my academic work. I depend totally on my students. So this is Haran Jyoti, who was a student in the 1980s. Chris Abel, of course, is a colleague, not a student. And then I went to another student called Steve Bunting, and got $500,000 from him to try out the ideas we had about making these drugs. And then after another 2 years we got 20 million investment. We've had 80 employees up on the science park, and we've got these drugs going into clinical trials. So my question is really what can we do apart from building new buildings? What can we do to do something about genetic disease? So, of course, what I do is I begin to look at the target, and we had lots of clinicians come and see us over the last few years and ask us about the fundamental rare disease that they're interested in. But I thought I'd just show this one. We published a couple of papers on AKU. I probably didn't know anything about it 2 years ago. There's a guy called David Asher, who's somewhere in the audience hiding, who's critical to this operation. And there's a computer scientist just here who came from Brazil and got a professorship rather sooner than I would have wished, and has disappeared back there. Well, we know what's happened to the mutations, and so the question is, how can we go from there? So the first thing we need to know is what the impact of all these mutations in AKU might be on structure. And it turns out that we can subclassify these by looking at their impacts on the protein. And so the way we do that is to use modern computer science, and I'm a biologist mainly, by the way, and we use what's called machine learning methods, and this came from the computer scientists. And we have test sets, and we teach the machine how to use this, and then use that from test sets to predict the effect of each of those mutations. And actually in AKU you can classify them into several classes, and now people are using those classifications which, for example, is the mutation between the subunits of the protein, and try clinical trials on each different classification of class. So then the question is, how do we go from there? So what I need to do, because I'm not going to be able to spend lots of money on it as we have done in Aztecs, we need to do some clever computer programs. So part of my lab does computing, part of my lab does experiments, and the other part tries to bring the two together in drug discovery. So we have a lot of different databases which take all the information we know about how a drug binds to a target. This makes models of the proteome. We then use the predictions to try and understand the mutation. And so we go through this process, and then what we try to do is to try and get from either this end or that end. And I'll tell you what these two ends are. This would be either a screen or a drug on the market, and over here you've got the target. And so the question is, if we have a drug on the market, we might want to ask, how do we get to all the possible targets that drug might hit? And so the other question we might ask is, we've got a mutated target, so we're down there, and we want to travel up here to get the best drug which is already on the market to treat. So we're using these fundamental processes, and I'm not going to try and explain them to you, to try and get an idea of which drug for which particular mutation might be useful to treat. And that's the process we do. And we use this sort of diagram for all sorts of other purposes. Like I say, we can use it for antibiotic resistance. A lot of problems we have in drug discovery is we do what's called a phenotypic screen. So we get a hit, but we don't know what the target is. So all of these methodologies we can sort of take from other areas. The Gates Foundation have been funding us, for example, and trying to do, I'll just go on what I've said. So what we're going to do then is we're going to understand the mutations here, we're going to go through that process, and then we're going to choose the drug which is most likely to bind, and then we have to do some experiments. And that's my story. See, I'm an academic.